Thank you. So yeah, I guess this is going to be a little bit of a retread for the folks that are in physics, but hopefully it'll be new for people that I haven't met before. So yeah, I'm what's that?
Biologist.
All right, great. So yeah, so just to give you sort of an idea of like where my philosophy is, let's look at two really beautiful movies. On one hand is a microtubule suspension, which I'll be talking a lot about today, so I'm not going to talk about the details just yet. And then on the right is a zebrafish embryo undergoing morphogenesis. It's a very complex orchestrated process. And I think that these sort of two systems are in some kind of conversation with each other where, engineering reconstituted systems provides us with a sort of simplified language for understanding more complex biological systems, while these biological systems also sort of inspire us to build more complex active materials. And so the idea is to, I guess I sort of see myself more right now on this bottom branch where I'm looking at biology for inspiration to make these systems more complex. So here's a little bit more information on the video you were just looking at. So we have microtubule polymers that have been reconstituted from living cells, and they have been suspended down onto an oil-water interface. And so this texture you're seeing is the sort of local alignment of these microtubule bundles. And they are made animate by motor protein complexes that are walking along those microtubules. And so in this very cartoonish schematic here, we have multiple kinesin motor proteins that are bound together with surcabidin, and this allows them to walk along multiple microtubules, and therefore creating this sort of like extensile motion in the microtubules. And I use the word extensile. How do we know this system is sort of stretching? Well, we can look at these FRAP experiments done by Linnea Lemma. And so the microtubules have been dyed, but they've been photo bleached in this square here. You can see that as the sort of texture of this material evolves, that square is getting longer, but it's getting longer along the direction of the microtubules. So this is how we sort of know that on sort of average, that the system is always sort of extending along the axis of these microtubules.
Sorry, could you just try to move? I don't know how to do it actually, but if you could move the zoom bar just so we can see your.
Oh God, yeah.
You click on more. There are options.
Let's see. Hide video panel.
Hide floating.
Okay. All right. Thank you.
Can they look at the surface area of the bleached region? Because you see extension and contraction.
Yeah. Yeah. So actually, Linnea did do that quantification, and so it's acting basically like it's an incompressible fluid. So it's area is conserved, which is cool. And then you actually confirmed it with a few different kinds of measurements. So both looking at the flow field and looking at the deformation of that element. Yeah, it's a great question.
It becomes chaotic, right? Base part will eventually go everywhere.
Yeah, and then it just they observe it for as long as they can, and it's exponentially getting longer for as long as they're able to observe it. Yeah. Yeah, and so that's a whole area of study. This is something that Kevin Mitchell and Linda Hurst at UC Merced have looked at, which is looking at the mixing efficacy of the system. And it seems like it's, especially in certain geometries, it's like near optimal, which is really interesting.
Yeah, so mathematically speaking, is this system a mixing system?
It is, yeah,
cool.
Yeah. Yeah, so the thing that's giving this system its characteristic texture is this bend, what we're calling a bend instability in the system. So if you were to use a externally applied flow field to align those microtubules and then look at the onset of instability, we see that it has like this characteristic buckling pattern that shows up. It's driven by that extensile stress. So you have a uniform system, you have some little undulation, and then the flow field created by that now deformed state feeds back and just sort of runs away to create this instability. What's interesting about the system is that those bend deformations saturate into topological defects. So these are like isolated regions of disorder. And what we're sort of seeing in the previous, what was sort of giving the characteristic flavor of the previous movie were the motion of these topological defects. So now if I label them according to their topological charge, so we have a -half defect, which are these like triangular objects, and a +half defects. We can see them sort of swimming together in the active dynamic.
You also have line defects.
Only in 3D, so it gets a lot more complicated there.
Because here what you show is, I mean, here the lines we see are defects, you said? Between the,
Which lines are you looking at?
the line of our fences to the,
Oh, like these are like crack things? Is that what you're saying?
Yeah.
That's just a artifact of trying to plot this thing. So we've got a pneumatic field which has this sort of head-tail symmetry, right? But when you use just like default quiver plot or streamline plot in Python or whatever, it always kind of freaks out because you have these sort of like baked in places where you part. We know how to get rid of those now, but so that's just like an artifact of plot. Yeah, that's not real for this system. Yeah, that's a great observation. Yeah, and so these defects get quantified according to their winding number. That's not going to be super important right now, but I think the main thing to point out is that each of them has associated with it, particular kinds of active flows, and so they interact with each other through this advection. The +half defects, because of their symmetry, tend to propel themselves, while the -half defects are still generating active flows, but in isolation would not have the net propulsion. So one of the grand challenges of active matter has been to try to corral this chaotic motion into other kinds of patterns. And so here's just a almost getting dated list of some of the really awesome experiments that have explored this. So these first two on the left here, I mentioned that the active dynamic is created by sort of pushing down microtubules onto an oil-water interface. And what's happening in these two is that underlying oil layer is not isotropic. It's been replaced by another liquid crystal. And that liquid crystal, because of its anisotropic friction, tends to guide the motion of the microtubules that are on top of it. And to make things more complicated, the one on the left, that underlying liquid crystal is being actuated by a magnetic field. So that's why it looks chaotic, and then all of a sudden you see this sort of like waning of the microtubules. It's because that magnetic field has been turned on. The underlying liquid crystal layer is aligning, and now it's easier for the microtubules to flow north to south. The strange geometry we're getting in the center here is because the underlying liquid crystal are forming these vertical focal conic domains, and basically they're circular domains where there's an easy way and a hard way for microtubules to move. And so the vortices are seeing lines of those circular domains of the underlying liquid crystal. And finally, this is work that was done at Brandeis and Touboul and done in Rodriguez's lab. This is the microtubule active dynamic that's been confined to a well, right? So now we're getting sort of circulating motion as a result of like physical confinement of the system. So, I mentioned that these motor proteins are bound together permanently in these motor protein complexes, and that's what's allowing us to have the generation of active stress. But recently, biochemists at Caltech, in collaboration with physicists here, have built a new kind of system where the motor proteins are no longer permanently bound to each other. They're still always walking along the microtubules, but they're only able to sort of exert relative motion between microtubules when they are bound together using light. And so there's a lot of fancy stuff going on here, but basically we're only able to create dimers for these protein complexes in the presence of light. And so now if you take that system and you pair it with the active pneumatic, you get really beautiful movies like this. This is work that was done very recently by Zara in the physics department. She just graduated with her PhD. And so on the left here is the light intensity that's being applied uniformly to the whole system. And on the right, of course, is the active pneumatic. And you see this like really sort of striking activity where the moment the light is turned off, the active pneumatic halts, almost immediately, and appears frozen. So yeah, of course, this is uniformly applied light, and things get even more interesting if you spatially pattern the light. And so here, only in the blue reticule is activity being generated for the system. You can see that the pneumatic is basically arrested everywhere else, but inside we have what looks like the sort of confined space that we looked at before, where defects are nucleating, circulating around each other. This is not as fun to look at, but this is like another case of another example of what you could do with this kind of system. So here the light has been patterned in the shape of this elbow, and it's a little unclear in these images, but what's happening is one of those plug tap defects that I said is like self-propelling is sort of following this track that's been laid out for it with the activity. And I guess my question that I'm interested in is, how do we sort of use these kinds of tools to systematically steer the system? So, one can apply sort of ad hoc patterns and just sort of, see what you get. But what I'm interested in is starting with a goal for the system, like a particular configuration for the microtubules, and asking, what is the kind of pattern that I should apply in both space and time that get me to that pattern? So I want to solve some kind of inverse problem, and I want to do that in order to sort of like pose that problem. What I first need is a model of the system, and so I'll introduce that probably like too quickly. But the fundamental field that we're sort of interested in developing equations of motion for is the pneumatic orientation. And so instead of just using a vector, we're using a second order tensor that is invariant to sort of flips in the sign of that vector. And we want to do that because our microtubule bundles don't really have a head or tail. They're not self-propelling along one particular direction. And so this is just a natural choice of order parameter for the system. And we can borrow from equilibrium liquid crystals the equations of motion for the Q tensor. So how does this rotate and evolve over time? And it has components that come from both the elasticity of this microtubule network as well as the flow. So E and omega are the symmetric and antisymmetric parts of the flow gradient tensor. And so this is flow alignment and rotation for that system. And we can maybe talk about details of that if folks are interested. The final bit, and the bit that makes this truly non-equilibrium, is that our Stokes equation, our momentum balance, is driven by the Stern divergence of alpha times Q. And this is our active stress. that gives us that buckling instability. Okay, so if we like throw this in our computer dish, this is what we get. We have this like active turbulence. And on the left, the little sticks are showing our microtubule orientation. The color is like the degree of order. So where order is low, this is where we have defects. On the right is our flow field. The color field is the vorticity, or sort of like the local rate of rotation for the system. And the vector field is the flow velocity. And in this system, there is a characteristic length scale that you can pull out using dimensional analysis called the active length scale. And it goes as like the square root of the elasticity of the microtubule network over the active stress strength, right? So as the active stress strength goes up, that length scale shrinks and you sort of get more defects closer together. And so in our case, the domain size is larger than that active length scale, but we can shrink the domain to be sort of on par with that active length scale. And then we get qualitatively different dynamics where we're no longer actively nucleating defects. Instead, we have just 2 +half defects that are sort of required by topology of the container that are just processing around each other. Okay. And so we're going to use this simple dynamic as our sort of first pass at a control problem for this system. And so in order to do, a control problem, we need a control goal. And so because of symmetry, we know that if we have one circulating solution, we must also have another circulating solution. And so we know that our system has sort of at minimum 2 dynamical attractors. And we're going to do a kind of control problem where we try to flip the system from orbiting, one of those limit cycles to the other one. And we're going to do that by, very much inspired by the experiments, by actuating that active stress field, so turning the light on and off. We're going to show this system a movie and try to get it to do something else. Okay, and so, any kind of optimal control problem, has to start with some kind of cost or penalty functional. And I'll just, sort of go through this term by term. The first is our state penalty. So this is penalizing the system for not doing the dynamic that we want. And in our case, we have a sort of a high dimensional trajectory that varies in space and time for our Q tensor that we already know. We've already run simulations, so we know what one attractor looks like. And we're going to try to make the system starting from the other attractors like look like that other trajectory. So we're penalizing it at every step in time. And we're going to also penalize the amount of active stress that we add to the system, and in such a way that we try to minimally perturb the system about its natural state. So we needed some activity just to get that circulating state to begin with. That's our alpha naught. And we're just going to sort of penalize deviations about that active stress. And finally, to help regularize the problem, we're also going to penalize gradients. And this also experimentally makes sense because we can't apply infinitely sharp gradients in light anyways. So we also penalize that. So we can definitely talk about this more if folks are interested, but I just wanted to lay out just the setup for this problem. We have our penalty functional that we're trying to minimize, but we're not allowed to just make arbitrary choices about how we do that gradient descent. We want to do that. We want to do our gradient descent in a way that respects the native dynamics that we've written out with our PDE. And so we're going to do something called the PDE-constrained optimization. And to do that, we introduce Lagrange multipliers that enforce our PDE operators. So the F sub U and F sub Q, these are our dynamics for the flow field and the microtubule orientation, and then incompressibility as well. And so we do something called Pontryagin's method to now find the optimality conditions for this system. It does get a little bit hairy, and we're not going to go through this thing term by term, but this is the PDE that you get for those Lagrange multipliers. So these Lagrange multipliers are not constants, they're actually governed by their own PDEs. And I think the key sort of like conceptual part is that these PDEs are driven by the error in our control. So it's like the system is not where it needs to be. That's going to be our error, our delta between Q and Q star. And that is like feeding into our Lagrange multiplier equations. And then we eventually use that information to update our control. And I think this is a horrible slide, but I'm just putting it up there in case we want to like talk about it later. Basically, we end up using a numerical approach called direct adjoint looping, which is basically this fancy gradient descent to find out what the space and time evolution of the active stress field needs to be to get us to our Q star. Okay, so now I'm going to show you the results of that. So we're starting out, and initially the system is rotating in the counterclockwise direction. And I'm showing this first time step. So already the active stress field, the control has been flipped on. So that's what you're seeing in this second row. And on the bottom is the instantaneous flow field. And so you can see that over time, if you just skip to the end, you can see that now it's rotating in the opposite direction. And what's interesting about the evolution of the director field over time is that in order to get from this, one-handedness to the other, we had to sort of pass this point of symmetry, where this, around time 0.72, the system is achiral, right? It doesn't have a handedness. And I think conceptually that sort of, makes sense. We had to go from one handedness to the other. We had to pass through a point that didn't have any chirality. But what's interesting about the active stress field that's being applied there is that it does have a handedness. And so we can see that what the control solution found for us is it moved us to this sort of point of symmetry and then kind of tipped us over to the other side to get us where we want it to be. And so this is, the control solutions that we get are sort of complex and need their own sort of forensic to sort of, I think, justify everything we see. But, the flow fields that we're getting are pretty simple. And I think the way to view this is that the flow fields are really in service of getting us to this symmetric state. I think we can look at how the pneumatic is being rotated. It's always driving us towards this point, and then the active stress field at that point just kind of knocks us over to the other attractor. Yeah, we can also look at these sort of net metrics of how the system is progressing. So the circulation, we can see when that changes sign. We can look at our overall control effort. We see that initially there's sort of a big push with the active stress, and then that tapers off. And we can also see how the system approaches where it needs to be over time. And so here's just a movie of what I just showed you in stills. In the upper right here is the control solution. In the upper left here is where the system is supposed to be at any given time. And then the corresponding flow and control fields. So I'll play that one more time. You can see that the target itself is a moving target, right? It's always evolving. And eventually we're able to bring the system into alignment. And so the control is sort of doing two things. It's both getting us to the other attractor and it's matching the phase. Yeah, Thomas.
In other applications, maybe as opposed to having access to this active stress, you might imagine situations where you could instead control the alignment of the pneumatic, or maybe even the fluid velocities.
Yeah.
So would the same kind of approach generalize if you had different controllers, and do you have any sense of which one is the most useful or the easiest to control the system to?
Yeah, that's a great question. I mean, this is one example. It's sort of like a crude way of controlling the flow. So that this is work that was done at Johns Hopkins, and what they've done is they have a magnetic disk that's floating above the nematic, and so you sort of see it out of focus here as this shiny blob, and so it's rotating, and eventually that sort of entrains the nematic to also rotate in that direction. So the problem can definitely be modified, and we did this control problem without knowing about their experiment initially, because it was sort of a natural thing to want to control. And so what we've done here is the vorticity tensor, we've just augmented it with an outside forcing. And so it's very easy to sort of set up different kinds of control problems. This is probably not, I guess what I assume in doing this control problem is that I have spatial control over the vorticity, whereas in their experiment, they really have this one source, and so they're kind of limited. So I don't think my control problem actually really matches their experiment in that sense. I think, I mean, the only way to really have individually addressable sort of pixels for orientation would be to have some kind of, I'm sure the experimentalists will have better ideas, but like, you know, magnetic actuation at some spatial scale. And I don't know what that would look like, but you need some kind of device where you can address individual sort of pixels. The active stress is the next best thing. We have like this indirect way of controlling the orientation, right? Because we create active stress that creates a flow that then reorients the system. So it is indirect. But yeah, it'd be cool to think about more direct ways of actuating things. And so here's just what it looks like for the vorticity. Yeah, I don't know, it works. And it kind of makes sense that it does, right? We use optimal control, but I think you can actually apply pretty naive guesses about what the vorticity field needs to be to get it to flip attractors. Okay, so this is a point that I kind of went over quickly, but it's maybe worth revisiting, that there's sort of like a lower dimensional way to think about the dynamics of this system. So we have, because of these two circulating states we have are two limit cycle attractors, and necessarily between them, we have this like unstable manifold. And so I kind of showed this earlier in the progression from one chirality to the other, that there is this unstable equilibrium state that doesn't have any net rotation. And actually there's a whole series of these states that all just like rigid body rotations of each other. And so we have like this line, if you can think about this system being projected to some lower dimensional state space, then we have a line of these unstable configurations in between two oppositely rotating limit cycles. And so one of the questions that excites me right now is trying to formally do this dimensionality reduction for this system. And there looks like there's some data-driven ways to do this. So we using the simulation data that we already have. Qualitatively, we know that this picture is correct, but maybe we could formally show it in some ways. But I guess what this also highlights is sort of the power of the optimal control solution that we found, which is that in this lower dimensional space, it's obvious that to get from one attractor to the other, we need to move in the z direction. There's no amount of moving in the XY plane that will get you from one attractor to the other. It will just change your phase. But in this like high dimensional PDE system, it's like not obvious what z means. You don't have a coordinate system that properly clarifies this. And so part of the exercise of finding the reduced dimensional model is to clarify what z means for the active nematic confined to a disk. What are the right coordinates to think about it, this system in? Okay, so this is going to sort of conclude part one of the talk, and I just wanted to say that yeah.
Changing of parameters. When you're going from one to the other, you're changing what the spatial temporal field. The alpha becomes a variable.
Yeah. So I am sort of like conflating control inputs and state variables a little bit. But I guess related, the simpler problem, like not even thinking about the alpha, but just to think about like what perturbations in Q get you closer from one attractor to the other is an important part of this dimensionality reduction. So like what kind of distortions in Q are most efficient at moving you towards the other attractor versus just changing the phase. You can like just make random twists to the attractor, to the system, and it's not super clear which it's going to do. It's probably going to do a little bit of both. It's going to like move you in some direction. If you can solve that problem, then it's more straightforward to figure out which kinds of active stress patterns do the same thing. Which kinds of active stress patterns just change phase, and which ones actually move you away from one attractor.
I mean, if you're gonna change topics, I have a few questions.
Yeah, go.
So my first question was about when you acted on the stress,
Yeah.
and was able to give the rotation. So I guess what you were doing was kind of project where you kind of froze where the rotation, where the singularity wanted to go. You kind of allow it to go the other way. My question was, can you always control actually, or do you have some cases where actually, the amount of light you need to put is too much compared to being away from the material? I mean, do you have a way to study that with the Lagrange multiplier and actually?
No,
you even solve the
yeah.
It's something I kind of like.
I don't know of a way to do this that's not empirical. So in order to find a control solution that works, I had to play around with like the weights on the penalties for these different terms. In a lot of the solutions, what happened is that it just gave up and it just didn't bother. It was like, this is better. The amount
It's
of
better to be away from my target
then expend the energy to like go to the other one. But I think there are some insights, in this like dimensionality reduction that can help us understand what the threshold amount of juice is needed. But yeah, right now in this PDE approach, the one thing I can think to do is like change weights and look at the kinds of control solutions I get and see when it's successful and when it's not successful. Yeah.
In this reduced dimensionality, are these like invariant manifolds separated into solutions, and then you're looking for gradients that are perpendicular, from one so that it goes to the other?
Yeah, I guess this plane in the middle at z equals 0 is invariant manifold.
Yeah, that's because of your symmetry.
Yeah.
So in that case, it's kind of explicit, but otherwise it's very hard to compute the fraction that's not for
Yeah.
But so now if you wanted to control that, like way, you would probably find the amount of energy you need to provide to cross the feed versus the cost it is to be away from the that would probably tell you exactly.
Yeah, I think it gets a lot more clear in this representation. And so I think we can also do the optimal control problem just on this toy problem to get some insights as well as to like what's at play.
I actually have a couple of equations you have up there. So if we take z equals 0, it looks like your fixed point is only at x equals 0, y equals 0. But I thought the z equals 0 was where there's no rotation and x and y are fixed. It's
You're saying at z equals 0.
right. So at z equals 0, that's the unstable solution where you have both where you have everything you don't have rotation, right?
Mm-hmm.
So I would expect x dot and y dot to be 0 if you don't have rotation.
Mm-hmm.
But your only fixed point in x and y there is at x equals 0, y equals 0.
Yeah, I see what you're saying. Maybe,
Does
there's a
X
typo in
dot
my
Y
text.
not map directly to the X and Y locations of the defects?
No, yeah,
Okay.
these are not, these are not defect coordinates.
Okay, so this is not defect coordinates, and then Z is the omega, or the angular velocity.
No, it's more
More abstract than that?
abstract than that. Yeah, you can think of these as being, what is a good way to describe? Glad you asked that. Yeah, it's a totally abstracted thing where I just wrote a system that has two limit cycles. Yeah, that rotate the opposite way.
But I think the richness of that maybe would be some kind of phase reduction, I want to say, you know. Like maybe you have a way in a system with two limit cycles to map this back to an abstract space where you have two, just like we do a phase reduction through these isochron, you know.
I guess the challenge is that, you know, I don't know how to apply the phase reduction technique when we're trying to move from one limit cycle to another. Like the isochron thing is certainly, I guess it's valid all the way up to this point, and then there's like a discontinuity in that description.
That's right. So you would need to have like two isochrons, and because the isochrons are inside the attraction bassin of one fixed point.
Yeah.
Of one limit cycle length. Yeah. So you can map the whole thing into. So now you would have two maybe two leaves, or because I don't see them side by side. But that would be probably something where I don't know.
No, yeah, I mean, I think like there's analytical expressions for the Hopf oscillator isochrons, right? So we should be able to sort of apply that at least a little bit here. No, I think that's really interesting. Now, actually, this is a problem that Youngman Park and I are working on a little bit, at least on the data-driven side, to try to like do the mapping formally. Yeah, no, it's a great question. Yeah, any other thoughts on this part? Yeah, so I just wanted to add that, you know, now the challenge is to actually try to like implement these things, and there's a lot to sort of do to get us to that point. But that this is an ongoing effort that's happening in the Brandeis MRSEC with Seth, Mike Hagan, Aparna, and Pengyu Huang working on the sort of machine learning side of things. And I think I'm probably gonna have to go through this maybe too fast to be super useful. So I'm not gonna show the math so much, but just try to give you a flavor for the kinds of problems that I'm interested in. And so I just showed you this example of using an external field to actuate a pneumatic and make it do something that it wasn't going to do on its own. But a sort of parallel problem, right, is to think about how to make materials that spontaneously organize into patterns that we're interested in. So this is, you know, how do we make this material more sort of like morphogenic, right? It does this beautiful chaotic mixing thing, but what if we wanted to make it do something more structured? This is just a cartoon for like fantasy of like, yeah, can we make it do a defect lattice? This is a very, you know, non-equilibrium, far from ground state configuration of the system. Can we use the active stress in a structured way to get us there? And, you know, sort of towards thinking about where we can do some kind of engineering, like let's just like look at a kind of cartoon flow diagram of how information flows in the material. We have our Q tensor that describes the system. How that structure is shaped dictates the kind of active stress that you will have in the system. That active stress drives flow, and then that flow in turn rotates the director field. So this is sort of the bird's eye view of the dynamics. And you know, one thing we can think about is like that active stress, right? Doesn't need to be, obviously we showed with the control that it doesn't need to be a spatial temporally fixed field. It can vary in time, but what if that active stress field were being controlled by some other process? And obviously this is what's happening in biology. Everything is very sort of like well orchestrated. You don't have everything on all the time. you have things being inhibited so that you only create stresses and things like where you need them to be. But I think that this, reaction diffusion idea can be broken down a little bit further into different steps. And I think these steps are broadly sensing and actuation. And so sensing is where you take, structural information about the system and you try to convert it into chemical information, right? So maybe the analog in biology are proteins that sense mechanical stress and they change chemical activity as a result of that active stress. Okay. And so this is sort of where I got hung up in trying to think about how to bring more control to these materials. I think there's a lot of things you can do to just have a reaction diffusion process that's happening in the active pneumatic that shapes its activity. But what about, how do we really like make molecules or think about things that do sensing? And the inspiration for thinking about this question came from hydromorphogenesis. So a hydra is a really fancy active pneumatic in the sense that its body has pneumatic order. So this is what you're looking at is GFP labeled actomyosin network. So it's like all this organism is sort of like one big sort of muscle. And what's interesting about it from a sort of physicsy point of view is that because we have this pneumatic texture on this sphere, we have constraints on the topology. And what's interesting is that the sort of key features of the hydra organism, the head, mouth, tentacle, and foot, are associated with topological defects in that texture of the actomyosin network. And what's been proposed is that these defects are somehow involved in the morphogenesis of hydra. And I don't know how true that necessarily is, but it's an interesting question. And one reason why the hydra is studied so much is because its body plan is extremely robust to like really large, I was gonna say excursions, but I mean, basically they slice these things up and they still grow back into hydra. And so from like a dynamical systems point of view, you can think of the body plan as being a dynamical attractor, and you're making these really big excursions in state space, and yet it's still able to find that body plan. And the question is like, what is the origin of that robustness? And so just the idea of having, defects being related to morphogenesis made me pose a problem that may or may not be, biologically relevant, but the idea is I wanted to know, like, can you make a simple sort of reaction diffusion system that is sensitive to these topological defects? How do you turn this like structural information into a chemical signal that you can then do other things with downstream? And so the idea is, again, how do we connect the mechanical state or the geometry of the system to its to chemistry in some ways? And I guess I want to be aware of the time. I should probably like wrap it up like right now.
10 more minutes.
Okay, 10 more minutes. Okay, thanks. So I'll just give you the punchline, which is that if you embed within this pneumatic a sort of like exotic but maybe still plausible enzyme, you can get this behavior. And so what I want you to imagine is that we have rod-like particles that are aligned with the pneumatic, but because they're embedded in the pneumatic, they're getting deformed by it as well. And so if we say that these sort of exotic molecules, as they get bent, they become chemically active. And the way in which they're chemically active is a paired source sink dipole. And so this is, you can think of this as being an enzyme where one product is being produced on the South Pole, and then that product is immediately getting eaten up at the North Pole. And so that seems kind of funny, but this is a enzyme that's sort of in permanent churn, right? It's always, using, going through fuel in order to create this this chemical gradient. But what ends up happening is, we have a system that's very analogous to what you would see in electrostatics, where you have dipoles, and gradients in those dipoles can give you effective charge. I think this will probably not go super well if I rush it. So I'm just going to sort of say that, by looking at the geometry of defects, we can write some fairly simple PDE governing equations that will tell us how these dipoles both align in the nematic and create chemical activity. And so here's just a simulation running those PDEs, and
how does the chemical interact with the pneumatic?
so the chemical does not directly
It just expresses according to
exactly
aligns to it and expresses more or less. Okay.
So I'm not showing the pneumatic directly here, but I showed the defects. And so what's happening is if I have bend, the molecules align relative to that bend, so perpendicular to it. And then as they're bent, they become more and more chemically active. And because of the way that the director is shaped near the -half and +half defects, the south or north pole of those enzymes will be facing away or towards the different defects. So they get the polarity of the defects becomes apparent. And the concentration fields that are created then corresponds to the minimum and maximum of those concentration fields created by those dipoles, then corresponds to the cores of the defects. And I think this is sort of like the best sort of like cartoon representation of that. There were sort of bends in the director field along this way. They've aligned in that direction, but the south pole is always towards the +half defects, and the north pole is always sort of pointing towards the -half defects. So, way too fast to be going through that, but I think the point is that we can use, the chemistry and geometric anisotropy to do some kind of sensing. This is really interesting to me because we've gone from this like tensorial topology of the defects to a scalar field. And the fantasy is that now that we have a scalar field as sort of a toehold, you can attach other kinds of chemical processes that then feed back on the active stress of the material. Now the question is, is it possible at all to make this kind of enzyme? And I think a combination of DNA origami and protein engineering might be able to tell us a little bit about that. But I'm also curious in learning more about the biology and just seeing what else is out there that might already be doing things, similar to this. Maybe not identical, but adjacent.
So it's fair to say that what you're doing is expressing molecules near defects so that if you read out these molecules and act on them, you will be able to control automatically
Yeah,
the system.
yeah.
Without having to track the defect and act on them.
Yeah, exactly.
Yeah, we'll make a fruit fly. When the fruit fly doesn't use, somebody doesn't shine light on the fruit fly to tell what to do. The fruit fly just makes fruit fly.
Thank you. You had a question?
Yeah, I'm just obviously I'm still learning all the math, but the biology in me is super excited about all of this because we do have the ability to mess with the folding of enzymes to figure out exactly where the binding is. You can do that. So yeah, I think you can make it. And just want to put that out there.
Well, that's the most optimistic.
I think you can actually do that.
Excellent. Yeah, thank you.
Yeah.
In terms of this question of converting the tensor into some scalar field, is there, I think what you're saying is that there's not a way to kind of do this as just a direct readout. For example, if you were just to take the bending of the pneumatic field, would that give you a similar kind of scalar field, or are you missing something?
You're missing the orientation, but the bending part of it is buried in here. So that's a prerequisite for creating the chemical activity, and it's just you need to also align the thing so that you can differentiate between the two defects. If you're just finding high bend, I mean, you can do other things, but wouldn't necessarily be able to tell the difference between the two defects.
But it would be possible to construct a similar scalar field from basically, maybe you need also the velocity field or with, I mean, you have some information about orientation just from a snapshot of the current state variables, right? So, I mean, I think my only comment is that maybe there's a shortcut where you don't actually have to solve like a chemical advection equation. You can just take information directly from the fields you already.
Yeah, no, I think it just depends on what you're after, right? I think, yeah, if you have global information, there's a lot you can do. I think the idea is that like if you're embedded in the material and you don't really have a lot to go, you know, if I'm just like one of these molecules, I'm trying to figure out whether I'm near a defect or not. Very restricted.
Here you're solving an evolution equation for the concentration. Is that right?
Yeah. Maybe we'll talk more about it. Yeah.
So here we are trying to build a scalar field by band information. I suppose because in the extensile system we have the band split. Does it mean that if you want to detect the defects in the contractile system, you need to build another scalar field by the split?
Yeah, and actually, but I think you could have done what I did based on display instead. I think you just have to envision a different cartoon for what the enzymes are shaped like. But I think that this would still work. You just, instead of activating where you have bend, you activate where you have splay. And then maybe instead of being oriented along the pneumatic director, maybe they're oriented perpendicular or something. Yeah. Yeah, I think it can work either. I don't think it necessarily has to change whether the system is active or whether it's extensile or contractile. This only cares about is what it looks like, what the director field looks like. It doesn't care about where the activity is.
Have any more questions? Let's thank Mike.
Thanks a lot for the question.